So hi everybody, thanks for coming out in the rain. I'm actually usually traveling in July, which is why it's been a couple of years planning. But January is the month that we don't teach at MIT. So here we are, and plenty of time to lecture and spend the evening with you all. So I'm going to be telling you about our work on the liver, which I think is an underappreciated organ. It's one that we don't think about a lot. We think about the heart as a pump, the kidney as a filter, but really the liver is the factory of the body. It provides over 500 different functions in 4 major different categories. So it does detoxification of drugs that we take in, as well as hormones in your body. It synthesizes proteins like clotting factors. It's responsible for energy metabolism, so fat, sugar, and protein metabolism, and it produces bile for digestion. So just to start out with a question, what do you think happens when the liver fails? What would you observe in a patient coming in with liver failure? So buildup of toxins in the bloodstream from the lack of detoxification, so that can lead to mental confusion and eventually what we call hepatic coma. What else? So lack of protein synthesis, so the lack of liver-specific proteins like clotting proteins lead to things like elevated bleeding time. So if you cut your finger, it would keep bleeding. The bile doesn't get dumped into the gut. In fact, it backs up into your bloodstream. So a classic sign of liver failure would be what we call jaundice or a yellow appearance. And then the energy metabolism would be wildly perturbed, and that's often why you would see a patient. So why would livers fail? Liver disease impacts about 500 million people worldwide. This is mostly driven by viral hepatitis, so hepatitis B and hepatitis C. There are other reasons for liver failure here in the developed world. Alcohol is very common, drug-induced liver injury. You all have a very potent actual hepatotoxin probably in your drug cupboard. Anybody know what it is? There's a black label warning. Acetaminophen, Tylenol. Very potent hepatotoxin. So don't take it for a hangover. First lesson of this evening. Okay, so liver disease impacts 500 million people worldwide. And basically, this is the reason I've studied the liver for so long is that this is an organ system for which the only treatment really is whole organ transplantation. So we don't have supportive therapies, as is the case for many other tissues. And whole organ transplants, as you know, are in limited supply. So when you give a liver transplant, you need to do what's called a beating heart donor transplantation. So that means the patients that come in that are providing the donor organs essentially are brain dead at the time of harvest. So it's a very limited set of patients that are available to provide these transplants. So much so that now the surgical community is doing what we call living-related transplants or split livers. So an adult can give part of their liver to their child, or you can take one donor organ and split it into 2. So that basically gets us twice as many, but still nowhere near enough. So there's about 40,000 deaths in the US every year from liver failure and about 3,000 to 4,000 organs. Okay, so what can we do about this if we don't have enough organs to transplant? So people have been thinking about cell-based therapies for this disease for some time, and they come in a variety of flavors. All of them take the hepatocyte, which is the main cell of the liver that provides these 500 functions, and try and use them to support patients. So one thing that you could do is just do hepatocyte transplantation. So you could take liver cells and inject them intravenously and try and encourage them to go home in on the patient's diseased liver and repopulate that liver. You can try and make dialysis-like machines that instead of just filtering blood like it does for kidney failure, they would house living cells, and that those cells would process the blood that's coming through. You can even breed animals that have humanized livers. So this has been attempted, for example, with pigs. And then you would have another source of transplantable tissue. And finally, you could engineer implantable tissue or make tissue engineering, implantable liver construct. And this is what my laboratory works on. So for all of these approaches, you need basically this hepatocyte that does these 500 functions. And the challenge for the field has been this piece of data. So if you take a donor organ and you take out the hepatocytes, you try and use them in any of these settings, and you put it in contact with plastic, so you culture it in the lab and you just look at one function, this happens to all 500, over time, this is what happens. So from the moment you get it in culture, it basically stops being a hepatocyte and it goes on to die. Okay, so these cells are exquisitely sensitive to their microenvironment, to the environment that they saw in the body. Once you isolate them with enzymes and put them in culture, this is a challenge. So this is what we've been focusing on in my group, and my computer got bored of me talking, apparently. There we go. And our approach might surprise you. So I'm an engineer by training, is to take these technologies that came out of the engineering community, the computer chip industry, that have made our computers so much faster over the last 50 years. So this technology, which I'll show you how it relates to the liver in a few slides, is called photolithography. And it's basically the patterning of surfaces, of silicon surfaces, by light-based methods. So you shine a light on a plastic-coated surface, and it allows you to create a computer chip. And this process has gotten better and better at finer and finer resolution over the last 50 years, so that we can now fit about 1 billion transistors on the same footprint that we could 50 years ago. So the material science community has been really advancing this technology. And what we like to do is basically borrow this for medical applications, in particular for liver applications. So I'm going to pass this around for those of you who've never seen a computer chip. Maybe at the end of the lecture we can accumulate all my props in the back, because there weren't coming. So this is a piece of silicon that's been micropatterned with a photolithographic technique. Okay, so we are not the only people out there thinking about how to use these microtechnologies or microfabrication technologies in medicine. And just to sort of step back for a second to say this is actually a huge field. So you can see folks working on what people call lab-on-a-chip devices, where you fabricate plastic parts instead of silicon parts like that. And you can do whole laboratory functions on these little chips. So this is actually a PCR machine on a chip. At a smaller length scale, as these technologies have gotten better and better, you can print DNA and proteins. And these are some of the types of technologies that we find here at the Broad, where we also now can print small molecules. So we can take all of the same kinds of printing techniques and actually print these tiny little microarrays. And then these technologies, as I said, are getting better and better and approaching smaller and smaller length scales. Another interesting trend in the community is a set of technologies that has come out of the nanotechnology community. And these actually are what we call bottom-up methods. So these are top-down. We start with the substrate, and then we pattern it, and we etch it away. These are bottom-up. And so this is a picture of a quantum dot, a nanoparticle material. This length scale here is about 2 to 5 nanometers. These are really interesting imaging probes. We use them in our work here for doing microscopy. You can use them in animal studies for all kinds of applications. They're very bright fluorescent probes. What's really interesting about materials at this length scale is that they change their properties depending on their size and shape. This is not really intuitive. You think about, if you take a brick of gold, it will look gold to your eye. If you shrink down that gold to about 100 nanometer particle, it will look red. If you elongate it into a rod shape instead of a spherical structure, it now becomes an antenna for near-infrared light and can be heated remotely. So these materials actually, the physics of them changes at this length scale, and so in the community, people are really interested in incorporating them in all these systems. So between microfabrication and nanotechnology, now we have this great suite of tools to manipulate things at a pretty small length scale. So what about the liver? I told you the liver was so important and the cells are unhappy in culture. So it turns out that this is what the architecture of the liver looks like. If we zoom in, so the length scale here, this is about 1 millimeter, and this structure repeats over and over in the liver. And these brown cells are those hepatocytes. So you can see they are in this gorgeous architecture in the body. If we zoom in on them, they have about 5 different other cell types that they sit near. They form nice connections between each other. Here in this space, it's actually not a space, it's a misnomer from the old literature, the space of Dis. This is full of extracellular matrix molecules, so scaffolding proteins. So the point of this is that this architecture of the liver is really precisely defined at these very small length scales, and this is the environment that we perturb when we take the cells out and we put them in culture. So all of the technologies that I described to you earlier are actually wonderfully poised to take this cell and now add back aspects of its environment and ask, how can I make it happy again? And how can I use it therapeutically? Okay, so what I'm going to tell you about is just that in 4 parts. So I'll tell you about how we use these tools to figure out how to make the cells happy. We call that a structure-function study. Then I'm going to show you that we did this with human cells. And when we were able to make human cells function well in the laboratory, it turned out that we could use them to study human diseases in a petri dish. Because the human cells are really different than rodent cells or animal cells of other species that we commonly study things in. And so this gave us a window into human diseases like the drug toxicity I talked about earlier. Then I'll show you how we're trying to take these now happy cells and make implantable constructs, so tissue engineering. And then finally, I'll touch on how on earth we're going to get enough cells to actually make these implantable livers. So the liver actually contains about 100 billion hepatocytes. And to have a therapeutic impact, you probably need at least 10% of them. Okay, so we need billions of cells. So where are they going to come from? Okay, so starting with structure-function relationships. So this is the piece of data that I told you about. So these are the cells, hepatocytes in culture. They're losing their function. And for those of you who like to see pictures of cells, so this is what they look like when we first put them in culture. So these bright spots are the nuclei. And they are what we call epithelial. So they're a particular cell type that likes to sit next to each other. And over time in culture, they spread out. They take out this scrambled egg appearance. And this is a mesenchymal phenotype. So this is what we call an epithelial to mesenchymal transition. And these guys no longer behave like hepatocytes, and they actually eventually detach and die. Okay, so this is what happens when you isolate them from the microenvironment. So we did 2 studies over the years to see if we could recreate a microenvironment where they could be happy and then use that. So the first is where we applied the photolithography technique that was used on that silicon substrate going around the lab to pattern the cells on a surface. So normally I showed you they were randomly dispersed. And in this picture, the red cells are the hepatocytes. So they're randomly dispersed, or some are alone, some have neighbors, a couple neighbors, and so on. So we patterned them by patterning glass now instead of silicon. And instead of patterning it with metal, we patterned it with sticky chemicals, sticky extracellular matrix molecules, collagen, that the cells would stick to. So we ran that same photolithographic process across the street at MIT, patterned glass with collagen stripes, and now the cells stick in stripes on the surface. And it turns out that this organization improves their function. So just being next to each other made them happier. On top of that, we added these green cells. So these green cells are supportive cells. They don't have any liver functions. Their main function in this system is basically just to support the hepatocytes. So we micropattern them and we co-culture them. And these 2 manipulations, micropattern and co-culture, stabilize the cells in culture. The next thing we did was try and figure out how long these had to be next to each other. So we did that by making, this was actually an experiment that was inspired by standing in the grocery checkout line, where they had these, you know those little checkerboard puzzles where you have to move the pieces around and create a certain pattern. So they had a part like that, and we started thinking, why can't cell culture be like this? Why can't I just move this cell next to this cell, and then after a while I could reconfigure it and move another cell next to another cell? Why do I have to just put them down and have the whole experiment go as is? We started thinking about how can we make a cell culture system that's reconfigurable, where I could ask what happens if I bring the cells together and apart, or bring in another cell. So in order to do that, we did another microfabrication manipulation, which is called MEMS. MEMS is microelectrical mechanical systems. And this is the evolution of microfabrication in the silicon industry, where people started making micromachined parts. I have a part here that we made this way. And what you do is you take silicon, and now instead of just patterning on the surface, you also subsequently etch it away. So first you pattern the surface, and now you etch away all the deep protected parts. And it allows you to make something that has micron scale precision. So you can't really appreciate it by looking at that, but if you look in the microscope, it has micron scale precision. And what it allows us to do is to culture cells on one set of these interlocking combs and culture the supportive cells on the other set of interlocking combs, and then bring them together and apart and study how the rescue happened. So this was done by a graduate student of mine, actually a postdoc of mine, who's now on the faculty at UC Irvine, named Elliot Hui. And he had done his whole PhD in MEMS at Berkeley. And this is him now operating this in a cell culture hood. And the point of this video is to show you that this is normally, in a MEMS facility, you would operate this with a robot. The point of this whole design was to make something so simple that a cell culture person could just take the end of a pipette tip and click it together, and what we call the transmission ratio of this part would be so precise that the cells would be 80 microns apart. You wouldn't have to do anything magical. You just take the end of your pipette tip and click this chip together, and the cells would be together and apart at will. Okay, so using these techniques, this is what we learned. When we use micropatterned co-cultures, basically, whenever we co-culture the cells, they're always happier. So even if you randomly organize them, they're happier. But the amount of liver function you got out of the system really depended on the organization. And when we did it with rodent cells or we did it with human cells, we got slightly different answers. So I'll tell you the answer for human cells. Human cells like to be in clusters of this size, 250 cells of each other, surrounded by these supportive cells. Okay? That's their sort of empirical sweet spot. And when we do that, now they, instead of dying as they did before, or having a transient rescue as if they're randomly organized, now you can keep them alive for weeks to months. Okay? So now you have a system where these cells are much happier, and this is just one function we've looked at. You can't look at 500, but we look at many of the functions. Okay. So the other thing we learned with that dynamic part is that the way this rescue worked was that the cells needed to touch each other. So if we put them 80 microns apart in that little device, no rescue happened. They needed to touch each other. That was sort of the bad news, because we're sort of thinking about making an implant one day, and we don't want to have these supportive cells around. The good news was they only needed to touch each other for a day. After a day, you could separate them, and they could create soluble signals that the supportive cells would secrete, so secreted factors that would support the liver cells. So they touch each other for a day, and then after that, they could put out stuff in the media, and these two things together would support our cells. So I should say, this is, we're here at the Broad, we're doing a big project now to find out what these molecules are that are supporting our liver cells, because ultimately, we just like to add them into the system. Okay, so that's all I'm going to tell you about sort of what we know about how to keep them happy. Now I want to sort of turn to like how we might use this information. So the next thing I want to tell you about is disease modeling. So what's interesting about the liver is it's one of the organs that is most species specific. So human livers really vary very much from monkey, from dog, from rat, from mouse. And so there are lots of studies that are done in animal models that ultimately don't predict human clinical trials. A lot of it has to do with these drug detoxification enzymes that we were talking about. They're different for humans than they are for animals. So one thing we wanted to look at is if we could make these little human microtissues, if we could study drug toxicity in them. The reason we wanted to do that is what I alluded to earlier. So if you typically take a drug and you run it through a discovery process, it takes about 15 years, about $1 billion, and ultimately, this data is still true. 8 out of 9 drugs fail in clinical trials, and up to 1/3 of them fail because of drug-induced liver disease that was not predicted, even though this had been through animal studies. Okay, so what we did was take a third technology, where we took the photolithography process that I showed you before, and we wanted now to be able to make these happy microlivers, not on glass, but on plastic. We wanted to be able to make them in a standard format that anyone here at the Broad would do research on. So these are multi-well plates that can fit into all of our machines that all of the robotic fluid handlers are set up to handle. And instead of making chips like I've been passing around, we really wanted to have the microlivers in this format. So in order to get that to work, we had to print collagen spots of the right size, of those 250 cell sizes, in each of these little wells. You can see them blown up here and blown up here. So each one of these wells has about 40 liver spots surrounded by the supportive cells. And so in order to do that, we had to invent a different engineering technology, which I'm happy to tell you about later. So this is how they look. And now, to anyone here at the Broad, this should just look like a regular plate of cells. And we can feed it with any robot we want, and we can image it on a standard microscope. And so now they're happier, as I showed you earlier. And they form really nice connections between each other. And this, hopefully this looks to you like a chicken wire network. For any of you who've ever thought about going to medical school, when you open a textbook and ask, what does the bile network look like in the liver? It looks like a chicken wire network. So we were really thrilled to see these in our cultures because actually these are two-dimensional cultures, right? They're planar, and I just showed you that the liver is actually three-dimensional. So in spite of that, they're forming these chicken wire networks in sort of 2D. Okay, so the next thing we wanted to do was take a really hard problem that had sort of haunted the field and see whether we could tackle it. So this is a drug toxicity that is really infamous. So pholyuridine was an antiviral. It's an HBV drug, hepatitis B drug. And in phase 1, so in safety studies, it was actually found to be lethal. So 5 out of 15 patients actually died in the first weeks of dosing on this drug. And so clinical trial was stopped. When they went back to try and figure out how this happened, it turned out that the rat data, the dog data, and even the monkey data were not predictive. The drug looked completely safe in that setting. And so trying to understand further what happened, the investigators went out to study all different animal models, and it turns out that woodchuck, for this particular drug, was the only predictive model. So we wanted to see if our in vitro human cultures could have predicted this. And so what we found was when we dosed these over weeks, that in fact these human livers could predict this toxicity as compared to a normal human hepatocyte that wasn't stabilized in this way. So this was very encouraging. This is sort of a retroactive experiment. We've started now working with people who have new drugs that are going forward to do prospective experiments. So this is a story that comes out of a company that I spun out in 2008 called Hepatic, for full disclosure. And they worked with another company, Company X, who was doing a clinical trial, and the clinical trial was halted because of elevated liver functions. So that's a sign of some potential liver disease. So they dosed the cultures with these drugs, found out what was going on, changed the drug, resubmitted to the FDA, and then moved forward. So the excitement for me is that this is an invention that we've made, and we're sort of redesigning clinical trials and making them safer in real time. So that's a drug example. It turns out that you sort of think of yourself as an inventor for a second. What would you do if you had a human liver in a dish? What's the biggest problem that you could tackle? So we got really interested in liver infections. So it turns out that there are a lot of liver infections. I told you 500 million people worldwide, hepatitis B, hepatitis C, malaria goes through the liver on the way to the blood. These are all things for which actually it turns out there are not good model systems. So these pathogens will not infect a mouse, and there's only, for each of them, there's one cell line, which is a cancer cell line, that they will even grow in the lab, even part of the way through their life cycle. So we thought, oh, well, we have the authentic host for these infections. Maybe we could create models of these. So about 5 years ago now, we started working on these two forms of malaria. So malaria, as you know, causes about 1 million deaths a year. Plasmodium falciparum is the form that is more virulent, more dangerous. But Plasmodium vivax has actually been targeted recently by the WHO because it's more chronic. It can hang out in the liver in a dormant form, and it can recur months to years later. And what that means from a public health perspective is if you clear malaria from the environment and it's still hanging out dormantly in livers in the community, it can reactivate and your whole eradication campaign is for naught. So both of these have been of great interest in the community. Okay, so people are working on drugs to kill them. There are vaccine programs for both, and then of course controlling the mosquitoes. So just to sort of remind you how this works, in case you haven't thought about it. So there's a female mosquito that carries a parasite, bites a person, goes into the liver. This is a life stage that is clinically silent. Now it's growing in the liver cells, expanding 10,000 times, but bursting out into the blood, infecting erythrocytes, red blood cells, and going through this cycle of every 48 hours or so. So this is the time where patients would present with fever and the symptoms of malaria. The mosquito bites this person now, the parasite gets transmitted to the mosquito, and now a new person and the cycle continues. Okay, so the liver stage has become very exciting for the community because if you can stop the parasite at the liver stage, it's what we call a bottleneck in the life cycle. It's before that 10,000-fold amplification, right? So if you have drugs at that stage, it's much more likely to be effective than later on in the life cycle. And as I mentioned, for Vivax, there's actually a dormant stage of the life cycle there. Okay, so if we had a model of the human liver stages of malaria, we could understand the dormant forms that I told you about. We could screen against drugs in the liver stages. So as I said, this is a great stage to have drugs against because it's clinically silent. Patients are well. If you have a drug at that stage of the life cycle, this is before any symptoms. And we can make vaccine development better. So it turns out a lot of the most promising vaccines that are now in development are live attenuated vaccines. So they are living parasites that go into your liver, but then arrest. They never progress. And what they do there is raise an immune response. And so there's a lot of interest in the liver stage for that reason. Okay, so as I said, so there's no good models of the liver stages, and we wanted to see if we could infect and create a model in these little livers that I told you about. And in fact, that's what we did. So this is what this little guy looks like in our culture dish. So this work was done in collaboration with Scenaria, which is a malaria vaccine company in Rockville, Maryland, and then lots of people here at the Broad. So this is this little parasite. This comes out of the salivary gland of the mosquito and goes into your bloodstream. It's 10 microns long, this little worm, and it goes into the liver cells. So here we have it in culture, and I'm summarizing 5 years of work here. That was led by Sandra Marsh in my group. And essentially what we see, we're zooming in on the liver cells in these cultures, is this is actually the parasite growing. I told you it grows 10,000 times. It's lots of little nuclei growing bigger and bigger over the course of about a week. And if we take human blood, red blood cells, and overlay it into this system, this actually will burst out and infect the red blood cells. So that means we've captured the full liver life cycle of this organism. And this, we think, is the first glimpse of the dormant form. So if we take Vivax, which is the chronic one, you can see early in culture, there are these little guys. Some of them are growing bigger over time, but some of them stay little. They stay little and they hang out. So 3 weeks is as long as we've done the experiment so far. These little forms persist for 3 weeks. This is exactly what a dormant form is supposed to look like. No one's ever seen it in culture before. And we can't actually claim that it's the dormant form unless we can wake it up. So that's what we're working on now. But we're really excited because in theory, if this is the dormant form, we should be able to screen now for drugs against it. Okay. All right, so one of the first things that my team asked me when we started working on malaria is there a better way to do these experiments? So I showed you those little red dots. In the beginning, we would count them under the microscope. We would see how we were doing. So working with Ann Carpenter's team here at the Broad, now we have an automated way to do that. And it has all kinds of careful controls in it. So instead of counting these dots, we can do that in an automated way. And we can put drugs on the system and see drug kill curves. So this is what it looks like. If you add more and more and more of a drug, you see less and less and less of the parasite. And these are two very well-known drugs that are in patients. This is an old drug, so it's less potent. This is a newer drug, and it's more potent, and we see that in these assays. The other really cool thing about this experiment is you see this blue line here. This is the one tumor cell line that I told you about that the parasite can be grown in. Okay, so this is a tumor cell line that was cultivated from a patient in Thailand. And if you look at, this is a profile of all the drug metabolizing genes in these cells. So this is an experiment that the Broad helped us do. David Thomas and Todd Golub's lab helped us set this, this assay up. It's all 83 drug metabolizing enzymes. You just get a quick snapshot using this bead-based approach called Luminex. And you can see that these cell lines that people use don't have very much drug metabolism. We know that, and we see it here. And so what that means is this is a drug that needs to be metabolized to be active. So if we put it on these tumor cell lines that don't have very much drug metabolism, the drug is less potent because it's not as activated. But now if we put it on the liver cells that have a higher level of metabolism, you can see this whole curve shift over because the drug required bioactivation for killing activity. So what's exciting about this is that there are drugs in development which are known to require bioactivation. We think this will be a better assay for those. This is a drug that we got asked to test. It's about to enter clinical trials in August of this year. And this was really exciting because the community came to us and said, we know this has blood stage activity, but we want to know if it has liver stage activity. And we could show them that it did. Okay, so I told you about structure function. I told you about modeling diseases. Next, I want to tell you about this idea of making an implantable liver with tissue engineering. Okay, so this is my third prop here. This is something that's kind of all the rage now, but when we started 10 years ago, it wasn't all the rage. This is 3D printing. And what we've been thinking about for some time is, wouldn't it be cool if you could build a three-dimensional organ? So this is a process called stereolithography. There's a bunch of different flavors of 3D printing, and if we want at the reception, we can talk about them. This one is light-based. And it's basically a three-dimensional version of the photolithographic process I told you about earlier, where you shine a light on a surface and you get a pattern. This is a vat of a polymer, a plastic that's light sensitive, and you shine a light on it and it hardens in a pattern. And then you drop the stage and you make the next level and so on and so forth. So this is like a little branching tree structure that was made on a stereolithography machine. You can make all kinds of structures this way, as you see here. So, of course, this doesn't have cells in it, but we wanted to build an organ this way. So in order to get this to work, we needed our cells to live in a light-sensitive plastic. Okay, so that's not so easy. It turns out this was a whole PhD thesis by a student of mine named Valerie Lusang. And I think this is my only chemical structure, but the point of this is that this is the plastic that we worked with, and it's sort of like spaghetti. So they're intertwined strands of spaghetti that are stapled together, okay? So it's a polymer structure, and it's really inert. It doesn't interact with cells at all. But the way it works is you mix cells in it with a light-sensitive chemical, and when you shine light on it, it photocross-links, okay? And it sort of has, at the end of the day, a consistency of Jell-O, and the cells are like the fruit in the Jell-O. Okay, so a second PhD student, so another 5 years, tried to figure out how to make the cells happy in this light-sensitive polymer. And she made these livers. We call these our contact lens livers. They have about 1 million hepatocytes in them, and what she found is shown here. So if you take hepatocytes and put them in this material and you look at one of those functions, they stay alive, but they don't function that well. If you use the trick I told you about before, where you add another cell type, so you co-culture them, you get a little boost in function. If you now add amino acids into the synthetic polymer that can bind to the receptors, so this is an RGD peptide that can bind to an integrin receptor, you get a little boost in function more. And finally, if you add another cell that's present in the liver, the endothelial cells, which are the neighbors that were right next to that brown row of cells that I showed you, you get happy livers. Okay, so we have these contact lens livers. They're light sensitive, and now they function in the dish, and we'd like them ultimately to be implanted. Okay, so now what's cool is now you can play with light, right? We're engineers, we could make all different structures. So these are some of the different structures we made over the years. And this is a device that we made where we actually make little liver beads. So this is a microfluidic droplet generator. And you can see the cells are starting in here. They're coming out, they're budding through, we call this hydrodynamic focusing. So they're being pinched through this device. They come out in these little balls. When you shine light on them, they polymerize into these little liver beads, okay? We can make buckets of liver beads that are happy in their little microenvironments, and now we're working on assembling structures out of these beads, as well as 3D printing with some of the technologies that I showed you that are passing around. Okay, so they are light sensitive, but they're not organized. So I spent the first part of my talk telling you how patterning was so important, and organization was important, and architecture was important, and now I have 3 cell types completely randomly dispersed in this jello. So what about that? So we wanted to, so in spite of that, I should say, they, if we put them, this is I think my only mouse animal set of data, we put them in an animal, they persist, and they make, they function like human livers, and we know that because if we look in the blood of the rat, we actually can see human liver functions. Okay, so we can see human albumin in the blood of this animal. And so this is kind of our first attempt to tissue engineering. We implant them. In order to get this to work, what do you think has to happen? So you implant something, a contact lens, in the body, and now I'm showing you that I can find protein products in the circulation. It has to get vascularized, right? So it has to, what does that mean? It has to recruit blood vessels from the host, right? And those blood vessels have to hook up with the tissue so that secreted proteins from those cells can get into the blood of the animal. So we did a lot of work in this study basically to focus on that vascularization. But the cells were still randomly organized. So then we got interested in how we could organize them further and improve their function in these materials. And so Kelly Stevens in the lab came up with this technique. This is a molding technique. And essentially what she did was cast these polymers on surfaces that had topology, okay? So if you have a surface that has some topology and now you pour cells on it, the cells will go to the lowest point, right? And then you can seal it up and flip it over and do the same thing on the other side. And you can have a system where cells came in from both sides, but now they're in a planar structure, and you organize them in all different ways. So this structure, this is a pyramidal well structure, so all of the wells are pyramids. The liver cells went in on one side, and the supportive endothelial cells went in on the second side. But in 3D, they're close to each other in a very specified way. Anybody want to guess where we got this structure? Everyday object that has pyramidal wells in it? This is a bike reflector. So go home and look at your bike reflector. That's what it looks like. Okay. So when we do this, and again, I'm going to spare you the data, when we do this, the cells function a lot better because they're pre-clustered now in this way. So they're pre-clustered, the hepatocytes are happy, but the endothelial cells, right, they actually are the cells that line the blood vessels, right? So here, we haven't put them in a really optimal way for them to hook up with the host vasculature, right? They're nice and close to the liver cells, they're giving them good signals, but if you want to get rapid vascularization, we reasoned that it might be good to have the endothelial cells already in cord-like structures in our system. Okay, and so in order to do that, we did a collaboration with Chris Chen, who was at UPenn, and he just moved to BU. We're really happy that he's here in town. And what he did was print us the vascular network. Okay, so how do you print a vascular network? What he did was take a robot and print a sugar network. So at the NIH, our program officer likes to call these our cotton candy livers. This is cotton candy, so it's printed sugar. We surround them with liver cells. And what's the thing about cotton candy that you all know? If you just even put a little bit of water on it, it dissolves instantly, right? So that's what happens here. So we put liver cells around it, and when we put this in media, the sugar all dissolves away. We call that a sacrificial printing technique, okay? And ultimately then what you get is a place for the endothelial cells to grow in a network that you can hook up. And so this is an experiment where we're perfusing now, emptying out the sugar in this network. And in a second now, you're going to see we're perfusing, this is in the lab, with blood. Okay, so using now 3D printing to print the vasculature as well as the liver structures. Okay, and when we implant these in an animal, you can see, you might not be used to looking at blood vessel structures, but we specified these cord-like structures, and we put them in, and the green cells are human cells that we put in, and the red cells have come in from the host, and this graft vascularizes right away when we put it in days. And the reason they're all yellow is because the red and green is overlaying. So the cord-like structures start out human, and the mouse vessels come up to the graft, and then they actually form vessels of mixed character, right? Because some human cells and some mouse cells, and ultimately over time, the mouse cells are going to take over that graft. Okay. All right, so that's where we are with tissue engineering. We still have contact lens livers. We'd like to make them a lot bigger. And I told you one of the main challenges is where we're going to get the cells. So there's something special about the liver. It's an old myth of Prometheus. I don't know if any of you have heard this. But the liver is one of the only organs that can regenerate. Okay? And what's so interesting about it is in liver regeneration, it's the mature hepatocyte that divides. Okay? In a lot of tissues, for example, in your gut, we think about the tissue, the way tissue homeostasis works, the way tissues normally grow and function is through a stem cell compartment. Not the case in the liver. It's the mature adult liver cell that's dividing when it gets cues to regenerate. And in fact, there's a pretty famous experiment known as partial hepatectomy. So you can do this in a mouse. You can take out a lobe of the liver, come back in a few weeks, and the liver is back to its normal size. It's a fascinating arena of research, in part because it grows quickly and it also knows when to stop growing. So there's a whole field of organ size control. This is the reason the experiment that I told you about works in patients, where you take an adult piece of liver and put it in a child. The adult's liver grows back, and the child's liver grows as the child grows, right? It's because of this phenomenon. Okay, so we knew this, and knowing this and being worried about getting 10 billion hepatocytes, you might think, well, why can't I just make them grow, right? They are able to grow in the body. Why can't we get them to grow in the lab? And in fact, this is a huge field of liver research. People have studied the signals that make liver cells grow for about 50 years. And George Michalopoulos at Pitt is really the father of this field. Okay, in spite of that, nobody could get human liver cells to grow in the lab. And so we had an idea that went like this. Maybe the trouble was that the cells were dying all the time. People were trying to study how to make them grow, and I showed you that they were unhappy previously. So what if we stabilized them using our co-culture trick, and then did what you would do here at the Broad, just screen for chemicals that might make them grow? So that's what we did. This is the work of Megan Shan, so this is 5 years in a PhD thesis. And so what she did is do a co-culture in a teeny tiny format. So this is a 384 format. So this is even smaller than I showed you before, where she plates the liver cells on top of a field of supportive cells. And the reason she did this was that so the liver cells, if they wanted to grow, they would have room to grow. Because normally cells undergo what we call contact inhibition. So if they're too tightly next to each other, they actually won't grow. So we screened a library. For the Broad, this is actually a small library of about 12,500 chemicals. And we found chemicals that made our cells grow. So this is just an image of this. So hopefully you can see this is where she started. The green cells are liver cells, and after 48 hours, there's a whole lot more of them. And the red cells are the ones that are actively undergoing mitosis. So you can see that there's more of them, and they're growing. She did this there's a lot of donor-to-donor variability in human hepatocytes. People are different from each other. She did this in 10 different donors, and they worked in all of them. So here you can see this is quantified. They grow. They still function like liver cells. And then I told you about that experiment before. We want to look more broadly at all the liver functions, and you can see that all the liver functions are up, and they're still a lot better than those tumor cells that I told you about before, which you would expect. So we're really excited about this, and we're trying to grow enough liver cells to do some of our experiments. But one of our colleagues had an idea, which was, if you could get liver cells to grow, maybe those chemicals would be themselves drugs for patients that had liver injury. So you have liver injury, right? Some of your liver cells are dying, and what you want is for those liver cells to grow back quickly, right? And what we call these are hepatoprotectants. And so we wanted to do an experiment to see if some of these chemicals, these are what medicinal chemists would call like bad compounds. These are just starting compounds. We don't know much about them. They're not very potent yet, but we wanted to see, could these chemicals function in that way? So the fastest way to do an experiment like that is to take a model of drug-induced liver injury in a whole organism that a colleague of ours named Wolfram Gosling had set up over at the Dana-Farber. And that is a zebrafish model. So these are transparent fish, and they're shown here. And what we did was just, I talked to Wolfram about this experiment. He said, well, we'll just add the chemicals to the water and see if the livers grow. And in fact, that's what we did, and the livers did grow. So in this experiment, he has a version of this fish that has a fluorescent liver. You can see that here. So we add the chemicals to the water, and the livers grow. And then what he had shown a couple years ago is that if you put Tylenol in the water, these animals get drug-induced liver injury. Acetaminophen in the water gives you drug-induced liver injury. And so we did the Tylenol experiment, that's shown here, and we added our chemicals to the Tylenol-exposed fish. And you can see that they're surviving a lot better than the untreated ones. So this is sort of like a Hail Mary experiment, but we're really excited because now we have these chemicals that can make the cells grow in the lab, and they might also be drugs. Of course, we have a lot of work to do to make them better, but this is a promising starting point. Okay, so just to sort of wrap up, I've told you about how we can study the microenvironment of the liver, how we can model some human diseases like drug toxicity and malaria, what we're doing to try and make implantable livers, and how we're going to get enough cells, hopefully, to do this work. And hopefully, you can appreciate how all of these tools that are coming out of the engineering community are really so valuable for our community. And I would put forth that it's not just the case in the liver, but also in other tissues. They might be amenable to this approach. So I want to thank you for your attention. This is my group, all of our collaborators here at the Broad and elsewhere, and our funding sources.
Thank you, Sangeeta. That was really, really interesting. We have time for questions, and we have 2 roving microphones, so we'll start here.
I have a question. Did you screen recombinant proteins in addition to small molecules when you were asking those questions?
We didn't, only chemicals. We are doing, actually Steve is here, he knows about this work. We're doing an RNAi screen now, also in the fibroblasts, as well as designing a gain-of-function screen.
Can I ask a second question? Did you ask in that last zebrafish experiment if it blocked cell death or if it enhanced cell regeneration and growth?
No, and so those are both good experiments. And actually, the other thing that we're doing with Wolfram is he has a bunch of reporter fish. So there are 3 or 4 canonical liver regeneration pathways that we'd like to know if they're acting through. So hippo, Wnt, et cetera. And so that's what we're doing now.
Other questions?
Sorry to lapse into a technical answer.
Hi. How long do you think before you'll be able to do this with humans and actually get a functioning liver in somebody?
Yeah, that's a good question. So I don't know the exact answers. If you look back to the tissue engineering history, the tissue engineered products that are on the market typically are 8 years from in vitro proof of principle to large animal proof of principle, and about 8 more years from large animal proof of principle to FDA approval. And that's been the path for skin tissue engineering, bladder tissue engineering, cartilage tissue engineering, bone tissue engineering. So I expect we're kind of on that time frame. We're in small animals. We'd like to be large in the next 3 years. It's hard to tell. This is actually a harder organ than the structural ones that I just described to you. Skin, bladder, bone, those are things that really what you ask for them in a transplant context is mechanics. whereas here we have a lot of processing functions, so it's a little bit harder.
Whoops.
So you mentioned the size, the size of the micropattern that makes the cells happier is about 450 or 490
500
microns.
microns.
So did you? I saw like if I'm not wrong, I saw the bigger size was about like 4 millimeters.
Mm-hmm.
So did you try something in between?
Yeah, that's a great question. So we've tried, the way that experiment was done was to vary the interfacial area between the two cell populations by a log at a time. And so that's the way we got those time points. So that was actually my thesis work. And no one ever did those 5 points again. So there probably is actually a point between 500 and 100, which is, they're actually both pretty high for human. That will be better. And the reason we know that is if we look at the cultures, if we stain them for albumin or P450 functions, we can see that the cells in the center that are far from the edge are not highly functional.
Okay.
So we've actually thought about making more of like a donut configuration, because you could have cells, supportive cells in the center too.
Mm-hmm.
And you can make arbitrary patterns. So there probably are ways to improve it further.
Okay.
Mm-hmm.
So I have another question. For the microwells, the 96 wells, is that system a 2D system or a 3D system? Sorry, I didn't like maybe.
Yeah. So for the malaria, the 96 well system, that is a 2D system. Actually, I forgot to pass this prop around, and I'm happy to, but it is a 2D system. I'll leave it to you first. Sure. Thank you.
I was wondering, as it relates to the work that you're doing with malaria, and you mentioned that the liver being a place where it's a bottleneck. If the virus changes, do you, in terms of what I've heard about how fast the virus changes, and there's drug resistance, et cetera, so are there other types of disease models that show that once you figure out the bottleneck, then you don't have to worry about it showing up again in people who are suffering from the disease?
Yeah, so I should say it's not the only bottleneck actually in the life cycle. And so people are working, there are several bottlenecks in the malaria life cycle, and you can sort of intuit where they are from the picture that I showed you. The other one is transmission, right? And so there are actually people that work on what are called transmission blocking vaccines. So you might not make the patient better, but you'll prevent them from spreading. There's also interventions. There's an amplification stage in the mosquito, which is another bottleneck. And so the bottleneck strategy is sort of well-known, well-developed strategy in parasitology generally. In terms of evolution of the parasite, that doesn't happen mostly at the liver stage. That happens in the other life cycles. There's a sexual stage of the life cycle in the blood where a lot of recombination happens, and that's where you can get a lot of resistance. We have also modeled really rapidly varying viruses. So hepatitis C, this is an amazing year for hepatitis C. I don't know if you guys are aware, but 2013 is like the year HCV became cured. It's like a moment in time. But anyway, HCV is very rapidly evolving, more rapidly than HIV. And we can see some of that variation in our cultures when we model HCV. One further back.
Hi. Thank you for the lecture, by the way.
You're welcome.
I have a question. Since you said with this liver model with the malaria that it's the first time it's been able to successfully infect these cells and had the dormant stage, and then when they're exposed to the erythrocytes, you've seen them burst out of that stage and infect the erythrocytes. Are you going to be looking more into what they're actually sensing and how they're going from that stage to the burst stage and going intravascular?
Yeah. So those are great questions. Just to clarify, so the bursting through to the blood stages on the falciparum side, So that's not the dormant form, that just goes straight through.
Okay.
And the Vivac side is when we see those dormant forms. And so we actually are super interested in what they're sensing and what the cues might be. There's one thing that we've been doing is talking to clinicians who've seen Vivacs in the field, about 50 years ago. So it used to be that giving Vivacs was a treatment for neurosyphilis. I don't know if anyone out there actually remembers this, but anyway, it's true. It was in the 50s, and it was a way to give fever, and fever was a treatment for syphilis. So you would give Vivacs, which was drug-sensitive Vivacs. And so there are a lot of clinicians, older clinicians around town who have great stories about Vivax flares. So they'll tell you that patients, when they get co-infected with other liver pathogens like HCV, that their Vivax would flare, that different strains have different variations. So the North Korean strains will stay dormant for the whole year and wait for the next rainy season. And the tropical strains will come out every 3 weeks. So there's all these really interesting pearls. And so, of course, that makes us think, oh, what are they sensing? Is it circadian rhythm? There's a whole circadian rhythm literature. So we're really, really interested in modeling some of those things in culture.
Thank you.
When you do the multi-cell contact implantation into animals, do they metabolize drugs in those systems?
They do. So what we did in that paper was show there's actually a nice FDA guidance document called the MISTI document, which is a metabolite safety testing guideline. I mean, what it says is that if you give a new compound in a clinical trial to patients, if 10% or more of the area under the curve of a daughter compound had not been seen before, you have to stop the clinical trial and go back and characterize the daughter compound as its own entity. So what we did in that study, in that paper, was take some compounds that had been flagged as MISTI compounds and showed that you could have predicted that they were 10% or more area under the curve in a mouse.
All right. Are there any other questions?
Over here, sorry. I see you have funding from Stand Up to Cancer. Is that the idea of replacing cancerous liver cells, or how does your work relate to cancer research?
Yeah, that's a great question because my lab actually is in the Koch Institute for Cancer Research across the street. So the other half of my lab works on cancer. And we have no projects yet on liver cancer, which I can't believe. But the cancer work was actually using the same kind of approach to look at cancer microenvironments, which have a lot of the same characteristics: multicellular interactions, extracellular matrix, three-dimensional position. And in that grant, what we were trying to do is grow circulating tumor cells up. So harvesting them from patients and then controlling the microenvironment so that they would grow, and then we could do sequencing on them.